Hello everyone and welcome to today's live broadcast, Complete Workflow Solution from Gene to Protein. I am Soon Pham from LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar sponsored by Gibco, part of Thermo Fisher Scientific. For more information, please visit www.thermofisher.com/proteinexpression. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the green Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems hearing or viewing this presentation, please click on the support button at the top right of your presentation window or submit your question through the green Q&A button at the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the process to obtain your credits. Now let's get right to today's presenter. We're proud to welcome Dr. Michael Spring. Dr. Spring is a global product manager in the SynBio section at Thermo Fisher Scientific GeneArt in Germany. Michael received his PhD from the University of Göttingen, Germany. After his research work in the HIV field, he joined Micromet, now part of Amgen in Munich, as scientist in the preclinical development of bispecific single-chain variable fragment antibodies against cancer and inflammatory diseases. In 2003, he joined GeneArt and worked as senior scientist heading a manufacturing group before switching to product management. As a product manager, he is currently responsible for the protein expression and purification and directed evolution services offered to customers in case of outsourcing is desired instead of purchasing reagents and do it yourself. I will now turn it over to Dr. Spring for his presentation.
So, thank you, Zoom, for introducing me. Hello to everybody. Good morning, probably most of you. My name is Michael Spring, and I'm a product manager at Thermo Fisher GeneArt here in Regensburg in Germany. I'm happy to present the first talk today on options for your gene to protein workflow and how you can use Thermo Fisher reagents and services to maximize your success in mammalian protein expression and purifications. Your workflow for that. I explain gene synthesis and gene optimization and how the combination of gene optimization and our world-class expression systems can help you with your research. Basically, that's all for the introduction. Here's the agenda. I first start with the gene synthesis introduction and overview. Then I'm telling you some or giving you some details about a multi-parameter gene optimization, a mammalian multi-gene expression study that we did, and some molecular mechanisms of gene optimization. And then I'll come to applications and benefits for your workflow using these optimized genes and expression reagents. Then we will have a question and answer section, hopefully. Okay, so what are synthetic genes, right? Synthetic genes are double-stranded DNA constructs. They are synthesized de novo from digital data according to your specifications, right? You send us the sequence and we produce the sequence exactly as you send it and specified it. Synthetic genes can routinely made in lengths of up to 10 kb. We can go longer, but that's not a routine order then. It's then a specific order, but we can go longer for sure. And all genes are coming 100% sequence verified, obviously. If needed, we can optimize those. And I'll come to that in the next slides. So here in Germany at GeneArt, we are the competency center for writing DNA within the Thermo Fisher company. So the next slide is coming up here. This is a short overview basically of the gene synthesis gene to protein workflow actually. So we support full workflow from gene to protein, obviously, and we can do, for example, apart from gene or in addition to gene synthesis, we can do mutagenesis projects, so we can mutagenize your genes. We can do cloning of your genes, plasmid production according to your needs, and also, and this is what we're talking about today, mammalian and baculovirus expression of your genes. Now, we all know that the genetic code is universal. That means some or all organisms use different codons to encode the same amino acids, right? So let me see if this works here. So for example, an E. coli codon that is frequent can be rare in Homo sapiens and the other way around. And codon usage patterns differ in each organism, obviously. So if we want to optimize a gene, we have to use the codon usage of the host organism, the organism that the gene needs to be expressed in. And codon usage is not only the only factor that needs to be considered when optimizing genes, it's also many other factors. And these factors need to be weighted. So that means it is always a trade-off between codon optimization and RNA optimization, right? And this is a multi-parameter optimization, a multi-parameter challenge basically. And what we do at GeneArt during the optimization is we weight different factors, and if possible we use the best codons, but not in all cases. It might be better to use the second best or the third best codon when other important factors also come into play that are more important. And this multi-parameter challenge is solved by our gene optimizer, and we're using a sliding window approach for that. That means we optimize all parameters within a given window, in that we optimize the first codon in that window first, using all available parameters, not only codon optimization, as I said, I'll come to that later. And we also take the extended window into account to optimize this first codon. And once this is done, this is fixed, and the window slides to the next codon. And this is done throughout the complete gene. And at the end, you have an optimized gene ready. So the sliding window moves from 3' to from 4', from 5' to 3', and all possible codon combinations are tested, and then the best combination is fixed, and the window slides one further. And this can be done, or this is done in several phases with more and more relaxed thresholds to reach the best optimization approach. Now, using this approach, we did a multi-gene study in mammalian cells. What is important here is you can also optimize genes for expression in autologous settings, right? So because nature does not optimize for maximal expression, but for maximal regulation. And if you change that to maximal expression, you can also improve expression in autologous settings. And this is basically what we showed with this study here. We took 50 human genes representing the most interesting protein classes like protein kinases, transcription factors, ribosomal proteins, cytokines, membrane proteins, and compared the wild type and the optimized gene. And we did that all in an HAK 293 cells. So, and here are the parameters that I just quickly spoke about that we were taking into account, and that we are always taking into account when we are optimizing genes, right? So it's not only codon usage, it's the GC content, it's splice sites, it's poly A sequences, direct repeats, RNA secondary structures, and so-called killer sequences, implant sequences. So it's very important to optimize the RNA, the transcription, and the RNA stability. Right, this is important. And on the right side here, you can see these different parameters are all implemented in Gene Optimizer, and we are dealing only with the coding sequence. So we are optimizing only the coding sequence. Now back to the study. Doing all this optimization approach with the Gene Optimizer, we optimized, so we compared wild type and optimized gene, as I said, in HAK 293 cells, and we did that we did a triplicate transient transfections for the wild type and for the optimized genes, and compared that compared these the expression levels using an internal gene for normalization. And what you can see here is basically plots, and also depicted as a bar graph. We succeeded to increase the expression in most of the cases quite nicely. Sometimes it's only 1.8-fold, so to speak, and sometimes it's 14-fold. Sometimes it's it was possible to detect the optimized gene only, not the wild type gene. For example, for this IL-2 interleukin 2, where we really had no expression with the wild type gene, but we had considerable expression with the optimized gene. On the next slide, there's already the summary. That means 100% of all optimized genes did express. 86% of the optimized genes expressed better than the wild type, and only 10% of the optimized genes expressed as good as the wild type genes, and only 2 of these genes were a little less good expressed as optimized version versus the wild type version. And we had optimization, through the optimization, we had increase in expression yields of up to 25-fold. And interestingly, these two, we thought that might be a problem with the cells, and therefore we tested different cells. I mean, we basically tested CHO cells in addition. So here we have the first 3 genes that are depicted in this table, and ZNRD1, EMG1, and VKORC1, which is a membrane protein that didn't express well in HAK T cells, 293 T cells, as you can see. But when we compared that or expressed that in CHO cells, we could increase the expression a little bit with the optimized version. And also with insect cells, SF9 here in that case, we saw a nice increase in expression with optimized genes. So basically, that was the summary. Now, how is gene optimization working? I mean, on what level of the cell does it work, right? So if we look at, for example, the MIP1 alpha here, we increased the expression 3 times, and let me see, and we detected that in a commercial ELISA kit, and that is composed, that 3 times increased expression is composed on several levels, so to speak. So gene optimization works on several levels of the gene expression. And for example, this was set together obviously from 30% better de novo RNA synthesis rate, then 14% better mRNA stability, 20% better translation efficiency, and also in addition we saw 80% higher mRNA steady state levels in the nucleus as well as in the cytoplasm. And here's a geo picture of that. So the increased expression is, as I said, is working on different levels of gene expression. So it's important to optimize for RNA stability, not only codon usage, right? Yeah, then some people are asking, well, your optimized genes, do they have the same activity or do they lose activity? And we did several experiments to prove that, or to find a case where where optimized genes have less activity, and we didn't succeed. So actually, this is one example where we tested the activity of that's Janus kinase. So what we did is we purified the protein, eluted it from histag, from nickel anti-A, did a pull down on substrate protein beads, and here in that plot on the left side you can see that we used the same amount of wild type and of optimized protein in our test, because what we did is we added ATP, and the ATP activated the kinase so that the substrate could be turned over, and we tested, detected the active protein with an antibody against the phosphorylated serine in the protein. And you can see that with the wild type and with the optimized gene, we had the same activity basically. This was an example that optimized genes are as good in activity or do not lose activity compared to wild type genes. So to summarize that a little bit, to summarize the study a little bit, gene optimization increases overall expression success rate, as I showed you. It improves the expression yields in general, and it does not change the solubility or activity. What I did not show is it also allows to confirm siRNA phenotypes. So that's really a good tool to increase your project success, your expression yields, right? And the study is published, by the way, and it's free available via PubMed. Now, gene optimizer versus competitor, right? That's we did also compare the GeneArt optimized genes with competitor optimization, and here's one example. So we used the same approach. Basically, we normalized against the standard protein, and what you can see here down in this bar graph is that the wild type and the optimized gene are threefold. So I mean that we increased the expression threefold by optimization. And interestingly, competitors did even decrease the expression of the gene versus the wild type expression. And well, next on the next slide, there are two more examples, because the first one is the one that I just showed you, and then we have two other examples depicted here. So actually, the whole study showed that competitor optimization also increased expression versus wild type, but consistently, gene optimizer overall was better than the competitor optimization, because it's the only technology with consistently higher expression levels, and all competitors failed in the one or the other. So gene optimization using the gene optimizer overall outperforms all other optimization approaches. Now, how can we make use of that, or how can you make use of that? As I said, we are offering gene synthesis and protein expression, so the whole workflow from gene to protein, and we are focusing on insect and mammalian expression, so we don't offer prokaryotic and yeast expression. We focus on insect and mammalian, and we're using the Thermo Fisher systems, expression systems, because as I show you in some more slides, these also have advantages, and the combination is really what makes the difference. What should you put into a very good position in terms of protein expression yields. So this is a principal service. These are the principal service types that we offer from gene to protein. The first type is that you specify the culture volume that you want us to express and purify. And you can specify the cells, CHO or 293 cells, and the deliverable will be expression plasmid, obviously, and the purified protein from the culture volume that you wanted us to purify your protein from, and the documentation including comma, CGL, and Western blot. Also possible is a service that covers the specified protein amount. That means you specify the protein amount that you want to have, and it will be a guaranteed amount that you will be delivered with, but then the pilot expression service or pilot expression needs to be done before, because obviously all proteins are different and we have to find out how much work we need to do to purify your specified protein amount, and for that we need to do a pilot. The deliverables are basically the same, expression plasmid and the purified protein together with the documentation. And then we have this pilot project where we check for expression yield of your protein. So these are the principal service types that we offer, and this is an example workflow how we work. I mean, as I said, we start with gene synthesis. Once we have the expression construct ready, we transfect the cells transiently. We monitor the culture daily in terms of cell viability and cell density, and we find out the best point of harvest, and then we purify and document and send the results to you, and then you can confirm whether we should proceed or not. If you confirm, we start the scale-up, for example 1 or 10 or 20 liters, and we can obviously purify secreted proteins and intercellular proteins. We either work with the supernatant or with cell pellets. Then we can filter the larger amounts of supernatants and then apply that on the affinity purification column, because the first column is always an affinity purification like protein A or nickel anti-A. Sometimes that's good enough and we deliver the protein to you together with documentation, or optionally you can order a size exclusion, a second column. In addition, we can do several different analyses with your protein. We can determine the endotoxin level. We can do an analytical size exclusion, copy-like, gel electrophoresis, whatever. And it all comes with a nice documentation. Okay. This is an example for documentation. Not sure if you can see it, if it's large enough, but it will always have the full set of information, what cells we are using, what vector we are using, or it basically can be specified by you. What buffer, the buffer that we deliver the protein can also be specified by you. Then we have the standard documentation with an stained STS page deal, This is just a standard code for the concentration determination. In that case here, we have done an analytical size exclusion to see for this antibody the percent of monomeric antibody here, and it also was and also, you know, we run this antibody together with a set of marker proteins that you can see it runs according to what you need. Now we have done several hundreds of projects in the past, and one example here is, and I wanted to show you some examples of that so that you can see what is possible and what can be the outcome. This is basically customer data. So they sent us, they did the expression and purification and send us the data. This is what they had in-house with the non-optimized genes in CHO cells. They wanted to combine heavy and light chains to get different antibodies, and they didn't succeed basically to express it all. Only the one antibody was expressible with the wild type genes. Now, then they ordered optimized genes, and this turned the project into a possible project, because then they could express the combinations heavy and light chain antibodies nicely. And here you can see how the fold increase was. In this case, it was really impressive. So from nothing to considerable amounts of milligram per liter of protein, of antibody, basically, were possible in that case. So that basically, the gene optimization basically turned this project into a possible project. Otherwise, with the wild type gene, it wasn't possible. Another example is shown here. So this is also selected from a larger study. These are all antibodies, and the white bar here is the wild type gene from customer in-house data. And then we used an optimized gene together with the, or cloned into a customer vector and a freestyle system. That's the blue bar here. And then we used an optimized gene in another vector, in the pcDNA 3.3 in freestyle. And finally, we used an optimized gene in the pcDNA 3.3 and the XB293 system, which is more advanced than the freestyle system. And you can see that really the combination of optimized genes and expression systems makes a large difference. Another example is from another pharma customer. They wanted 2 antibodies to be produced, and they wanted 250 milligram of the antibodies, and in-house they had 2 milligram per liters, and they used wild type heavy and light chains for the antibody, and another leader peptide in the freestyle system. And we said, okay, can we please use an optimized gene and our leader peptide and our expression system? And they said, yeah, that's okay. Then we could produce the 250 milligrams already in 1 liter. So that really was a nice outcome, and shows really again the advantage of the combination of optimized genes and expression system. And the good thing is that all the reagents are commercially available, so you can order the gene here at GeneArt and use and also order the reagents and do the work in-house if you want. This is a comparison of XB293 and XBCHO, and also here we can see that, and I must say these are all optimized genes. So we used only optimized genes and wanted to see the difference between the 2 expression systems, and it turned out that really the XBCHO system could increase the expression of a low expressor antibody, a medium and a high expressor antibody to a certain amount, and so the system can, the XBCHO system can increase the expression, but that's not necessarily the case for all proteins, right? It's really target specific, and it needs to be tested for each protein. Now, quickly how to order protein expression services. Just go to the Thermo Fisher web page, type in GeneArt gene synthesis, and click on the GeneArt cell lines and proteins section, and then you can download a questionnaire that you complete and send to a specific email address, geneartprotein@lifetech.com, and then there's basically a project manager will come back with the code or with additional questions if this is not clear from the information in the questionnaire. So it's easy to order, and it's a personalized service. I hope I could show that we have several advantages for your gene to protein workflow, which is really we have the complete service chain, but you can also use parts of the service chain. But in case you want to use the complete service chain, everything is done in-house, from gene to protein, from the oligosynthesis to the purified protein. And we can be quick. It can be in 30 business days if it's only a standard gene, a short standard gene and a standard purification. We have a very reliable performance because we are using these advanced expression systems and in combination with the world-class expression optimization. We have a scalable and flexible service here, right? It can be from milliliters to 25 liters, and our team has experience with large projects, and we can customize your purification protocol. In case you want us to just purify according to our standard protocol, that's fine. If you want us to establish your protocol in our labs for larger orders, it's also good. So it's a flexible team, and it's a really an experienced project management team that is able to communicate in all phases. I mean, the ideal world is you order, then we do something and you get back the protein, right? But what is if any issues appear during the project, we will immediately call you and give you the opportunity to, and show you the data and what the problem is, and give you the opportunity to tell us how we should proceed. Okay, and that means it's a smart way to receive purified protein in short delivery time and the optimized gene in a suitable expression vector for your further in-house work. Okay, with that, I'm at the end of my presentation already, and I think we are now coming to the question and answer section. Just in case you have any questions, go ahead and I'll try to answer these.
All right, well, that was an excellent presentation, Dr. Spring. Thank you so much for bringing the information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us today. If you have a question that you'd like Dr. Spring to answer, you can click on the green Q&A button at the lower left of the presentation window and type in your question, and we'll try to get to as many as we can. So our first question comes from Muhammad at the University of Tennessee in Knoxville, who asks, is all of the gene optimization program free, and which one is reliable in your opinion?
I think the question is, all of the gene optimization program is free, which is reliable. I mean, I don't completely understand the question, but basically, I got it. So all optimization is free. That means you can for example, you can order. You can order genes basically by two different ways. First is you can use our online portal. When you go to the Thermo Fisher Scientific web page and type in GeneArt, you arrive at the GeneArt site, the web page, and then you can enter the portal from there and use this portal for optimizing your gene. And you're guided nicely through the optimization, so no worries. Don't just type in or copy paste your sequence and then follow the portal commands, and you will be able to optimize your genes, and then at the end, sure, order the genes. But the optimization itself is free. Next question.
Okay, if you have any more questions, please feel free to submit them through the green Q&A button at the lower left of your presentation window. We'll just give everybody a quick minute to type in their question. Okay, next we have a question from Ramez, who asks, what are the factors that can influence the efficacy of optimization?
So the next question is, what are the factors which can influence the efficacy of optimization? Well, I showed you some of the factors that are important to optimize, to have a good result of the gene optimization. Most important, commonly known is obviously codon optimization, right? Codon usage, adaptation, but also the GC content is very important, and also motifs to avoid that lead to destabilization of your messenger RNA. So it's important that the transcription is working nicely and quickly, and also that the stability of the RNA after nuclear export is good. And these are the most important factors.
Okay, next we have a question from Sherry, who asks, are these products available in China?
And the answer to the question, are these products available in China, is yes. These are available in China. We have a dedicated logistics team that sends all our genes and proteins to all customers worldwide.
Okay, if there are no more questions, I'd like to thank Dr. Spring once again for a fantastic presentation. Do you have any final comments for us before we leave?
Okay, since there are no more questions, thanks very much for your attention. I hope it worked nicely for you, although we had some technical issues. And I thank you very much, and I hope I could show that really gene optimization in combination with Thermo Fisher expression systems really can help you with your gene to protein workflow. Thanks very much, and goodbye.
Well, thank you again, Dr. Spring, for a fantastic presentation. I would also like to thank Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. And to let you know, today's webcast will be available for on-demand viewing through May of 2017. You will receive an email from us alerting you when it's available for on-demand viewing and posted on labroots.com. Please feel free to forward this announcement to any colleagues who weren't able to make it to today's session. Thanks for logging in and participating in today's live broadcast, and we hope to see you again next time. Bye-bye.